Welcome back, guys. In this video tutorial we will going to solve some problems from Meselson and Stahl experiment. So let's talk about the first question. So I have taken this question from the Pathfinder CSIR Net MCQ book. You can also refer this. So let's talk about the first question. So a bacterial culture growing in a medium containing 15 NH4Cl. So in the previous video where I have discussed about the Meselson and Stahl experiment, I have told you that we are using ammonium chloride as a nitrogen source for the bacteria, right? You switch to a medium containing 14 NH4Cl for 3 generations, resulting into 8-fold increase in its population. Okay, and they want from us about the molar ratio of hybrid DNA to light DNA at this point. So let's start to solve this question. So what they are telling us? They are telling us that first they have grown the E. coli in 15 NH4Cl medium. That means our DNA will be N15 N15 fast, right? Then we will going to transfer them in a 14 NH4Cl containing medium, right? And as a result we will get two DNA molecule. Each of them will have one N15 strand and another N14 strand, right? We will get two DNA molecule having this kind of composition. Okay, so this is our first generation, right, guys? Then after that we will get two hybrid and four light. How? Because understand that we are getting two hybrid after the ending of the first generation. So after the ending of the second generation we will get one hybrid, one light, and here again one light, one hybrid. Okay, that means two hybrid and two light. Clear? That is the ending of your second generation. Now after the ending of the third generation we will get one hybrid, one light, and from these two we will get, here we will get two, here we will get two. That means total four light, right? And here again one hybrid, one light. That means 4, 5 and 6. We will get 6 light and 2 hybrid. Now what they are telling us, what is the molar ratio of hybrid DNA molecule? So we are getting 2 hybrid, right, out of 6, because we have 6 light DNA. So our answer will be A. Understood? So that is the first question. Now let's talk about the second question, that is E. coli cells that have been cultured in a light medium. So I have told you that they can also change the entire pattern of the Meselson experiment. We have grown the we have grown the E. coli first in a heavy nitrogen medium, but here they are telling us that we have grown the E. coli first in a light nitrogen medium. Okay, although the entire process will be same, but it will be the exact opposite. Okay, that means here you will get higher proportion of heavy nitrogen and lower proportion of light nitrogen. Okay, clear? Now let's move. As the sole nitrogen source were transferred to a heavy nitrogen medium containing 15N. So what will be our outcome after first generation? Our outcome will be, we will be having two DNA molecule containing N15 N14 combination each, right? Now they are telling us after 3 generation of growth in the heavy medium, what will be the proportion of the dual light strand, dual heavy strand and hybrid light plus heavy strand in the DNA duplex. So now let's try to think about this, that first we have grown our DNA in 14N containing medium, and then we have transferred them in a heavy nitrogen containing medium. That means the proportion of the heavy nitrogen will be higher. There is no doubt about this, right? And if you have watched my previous class, then I think you can understand that after the ending of the first generation there will be no pure 14N containing DNA, right? We will get hybrid. Okay, so that means they are just want from us about the ratio of heavy nitrogen containing DNA strand and the hybrid nitrogen containing DNA strand. Clear? That means this option will be cancelled out. So we have to focus on these three. And here I have also told you that the proportion of the your heavy nitrogen containing DNA strand will be higher, right? So in if you focus on the first point, they are telling that 3/4 hybrid strand, but that is not possible, right? I hope you can understand. So your answer will be B. Okay, because in previous video we have talked about this, that after the ending of the third generation, the medium in which you are growing your E. coli, depending on the nitrogen source, 75% of the DNA will be having that nitrogen source containing nitrogen bases, right? So that is what I want to told you, that is that after the once we have transferred the DNA from 14N to 15N, and after the third ending of the third generation, there will be 75% of the DNA that will have 15N nitrogen in its nitrogenous base. Okay, so this will be your answer, B. Okay, that is how you can easily guess your answer instead of doing the experiment multiple times. Okay, and that is why I have told you to please remember the chart that I have provided in the first video. Okay, that will help you to remember or to easily solve the questions. Okay, so now let's talk about the third one. So this one and this one is quite similar. Here it is also a GNU question. So in the Meselson and Stahl experiment, E. coli cell grown on heavy nitrogen. So here they are telling us that E. coli were grown on heavy nitrogen containing medium first, then it was transferred to a light nitrogen containing medium. So I have told you that simply means that the proportion of the light nitrogen will be higher. So I have told you that if you are growing your DNA for a number of generation in a light nitrogen, so the proportion of the light nitrogen containing DNA will be higher. Okay, and as it is the ending of the third generation, your answer should be 75%. Okay, so that is how you can easily answer your questions. Now let's talk about this one first. This is the short question type. Isotope used for providing semi-conservative replication of the DNA. So it will be your number B. N14 and N15 we have used, right? Now they are telling us, let's talk about this question, that is E. coli cell grown on 15N medium are transferred to 14N medium. That means the DNA containing 14N nitrogen will be higher in number, right? And allowed to grow for 2 generation. DNA extracted from these cells is centrifuged. What density distribution of the DNA would you expect in this experiment, right? So what will be our composition after the ending of the first generation? We will get 2 DNA molecule each having this composition, right? So upon the ending of the second generation, we will get 2 hybrid, 2 light, right? So your answer should be D. Okay, so that's the video, guys. So we have today solved 5 questions from Meselson Stahl experiment. Now I think you can easily solve all the questions that were asked in various competitive exams and also in various MCQ books. So thank you for listening to this class.